Hello, ladies and gentlemen. Welcome to this third day of Pint of Science Belgium 2020. Unfortunately, also today we are here in our living rooms and bedrooms because, the conditions actually doesn't give us so much chance to do elsewhere. It's set for tomorrow, but we are going to talk about this later. Tonight with me, Michele, your host. There will be a new enter for the online Pint of Science team. Tanin.
Hey,
Hi,
how's
Tanin.
it going?
All good, all good. And you?
Chilling. Ready to
Good, good.
learn.
Yeah, I'm chilling as well. I have also my beer. Where is it? Yeah, my beer here, so it's perfect. So we are ready for this third day. Today we are going to talk about something very interesting. And as the previous day, we were able to put together a combination of two different topics that might seem not very connected to each other, but in the end, they are connected to each other in a certain way. We can say that. We can say that. So first of all, before to start with everything, just some reminders for our audience. So you can, and actually you should, write all your comments in comment session. If you are on Facebook, on YouTube, doesn't matter. It's the same. Please share the link for this live with all your friends, because the more we are, the better, because the conversation gets richer and richer. And what we are going to do tonight, if it's the first time that we are here, you are here with us in this online version, we are going to have two speakers. After each speaks, we are going to have a Q&A session where you can do all the questions that you want regarding this topic, or you can even do actually just regarding their career, if you like to. I don't know, maybe it would be the case. But after that, we are going to have, after the first talk and the first Q&A, we are going to have a small break of 10 minutes. And after that, we are coming back with the second one. This is just to give you the chance to grab another beer, because of course you should be on your couch enjoying this nice talk and drinking a nice fresh beer. Without waste more time, I just want to introduce our two guests for today. From the University of Liège, she's a PhD student and she works in the Department of Sleep and Chronobiology Lab. Daphne Chylinski. Hi, Daphne.
Hey.
How are you?
Great.
Thank you for being here with us. You will be the first speaker, and you're going to speak with us about the importance of sleep, even though actually I think this is quite a very, very summary and probably not very good, but probably you're going to go deeper in that. I'm sure about that. And the other guest, which is going to be with us tonight, is Sebastian Proust. He's a senior postdoc at KU Leuven and VUIB, and he works for the Center of Microbiology. Hi, Sebastian.
Hey, how are you?
Fine, and you?
I'm also fine, thanks.
We are so glad that you are here as well. In this second part of today, you are going to talk about bacteria and microbiology, but in a very interesting point of view, actually, that we are going to understand better how to know our own gut, actually, and our own.
So.
So it's very, very interesting. So again, for the audience, if you have any questions, just drop in the comment section. I don't want to waste more time, and I will just leave the stage to Daphne. Me, Tanin, and Sebastian, we are going on the backstage. We are going to listen, and we are going to be very ready for more questions. But we leave the stage to you, Daphne. Just give me a sign when you're ready with your presentation, and then we just leave you the stage.
I am.
Okay, ready? Perfect. So see you later. And as always, good luck.
Thanks. So good evening, everyone. I'm very happy to be here with you tonight, and I hope I will unravel a little bit some of the mysteries of sleep. But before we start, I'd like to make a little thought experiment with you. So I'd like you to relax for a bit, maybe close your eyes, take a sip of your beer, and try to imagine that scientists came up with a new magical pill. And this magical pill has some amazing properties. It helps reduce your stress. It makes you more productive at work. It reduces some health risks, such as the risk of having a heart attack, the risk of having type 2 diabetes, the risk of having a stroke, of developing dementia. It also helps you stay fit and reduces the risk of having mood disorders. Sounds a bit too good to be true? Well, actually, this is all what getting regularly a good night of sleep will do for you. To take things to the extreme, let's see what extreme sleep deprivation does to you. So in 1964, Randy Gardner, a student, tried an experiment and did not sleep for more than 11 days. That's more than 264 hours without sleeping. That was for a science school project. And some of the effects of sleep deprivation you might have already experienced, but others probably and hopefully not. And those include difficulties to concentrate, memory loss, irritability, but also some psychiatric effects such as hallucinations and even paranoia. Of course, that's when taking a really, really long sleep deprivation. So you might have probably experienced that. The longer you stay awake, the longer you tend to sleep the night after. And this is because of what is called the homeostatic process. So this homeostatic process can be visualized as an hourglass mechanism. It basically represents your sleep need. So the longer you will stay awake, the more sleep need you will accumulate. And then when you go to sleep, this sleep need will dissipate until you wake up, and so on. Of course, this is not the only thing that regulates sleep. Otherwise, we wouldn't experience things such as jet lag, right? So why do we experience jet lag? Well, because we also have what we call the circadian process, which comes to counteract, in a sense, the homeostatic process. So actually, we have all a clock inside of us that at some point during the day promotes wakefulness and at some point promotes sleep. So around the end of the day, when you have already accumulated quite some sleep need, your circadian system kicks in and promotes wakefulness to help you stay awake for a consolidated period of time over about 16 hours. And similarly, at the end of the night, when you have already dissipated most of your sleep pressure, your circadian system will send the strongest sleep signal to help you stay asleep until your wake time. So here you can see the two processes together. So what happens in the case that you don't go to bed for, let's say, 2 days and a night between that? So you keep on accumulating sleep pressure, and your circadian system just continues oscillating between moments where it promotes wake and moments where it promotes sleep. So actually, during a normal day, those two mechanisms work together so that you have almost constant performance, that you're able to concentrate more or less in the same way throughout the whole day. But when you don't go to sleep, you have already accumulated a lot of sleep need. And towards the end of the night, in the early morning hours, this is the moment where your circadian system is really promoting sleep. So this is the moment where most accidents due to human errors occur because of this promotion of sleep by your circadian system. Similarly, when you take a nap, you just simply dissipate some of the sleep need that you have accumulated, and it might be, and probably will be, harder for you to fall asleep in the following night. So why do we experience jet lag? Well, simply because our internal clock is on a different time than the time zone where we arrive. So it has been proven quite a long time ago that circadian rhythms are endogenous and not just to humans, but also to, well, I hope I don't say something wrong if I say that it's common to most or all living creatures. So this guy, de Meyran, noticed that there was this plant that kept on opening and closing its leaves with a rhythm of about 24 hours. So during the day, it would open its leaves and at night it would close them. So he thought, okay, well, this is maybe because just of the light exposure. So what he did is he put the plant in complete darkness for a few days. Well, guess what? It continued opening and closing its leaves with a rhythm of about 24 hours. So it was already proven in the 18th century that circadian rhythms are endogenous. And it has also been shown in humans quite some years later that circadian rhythms are endogenous in humans too. So some scientists took a bunch of people and put them in a cave, and they kept them there for quite a few weeks without having any output from the outside world. And what they saw is that even without knowing what time of the day or night it is and without having any daylight, well, people kept on sleeping and waking up with a rhythm of about 24 hours. And I say about 24 hours because in most humans, the circadian process is about 24 and some minutes long, so about 24 and a half, 25 hours long. And this is why, for instance, it is a lot easier for us to stay awake longer than a regular day than to go to bed earlier than what we usually do. So because of this, because of the fact that our circadian clock is slightly longer than the day-night period of the outside world, the rotation of the Earth, we need to constantly realign our body clock to the external world. And how does this happen? Well, there are many what we call Zeitgeber, so time givers, so cues from the outside world that help us synchronize. But the most important one is light. So actually, the master clock in our body is situated in the suprachiasmatic nuclei, which is located in the hypothalamus in our brain. And it actually receives input from our eye. So in our eyes, we have some receptors that are dedicated to image perceptions, but we also have some specific receptors that solely transmit light information. And light is very important because it has arousing effects, antidepressant effects, and exposing yourself to bright light in the morning helps you recalibrate your body clock to the outside world. What happens is if you expose yourself to bright light in the evening, well, you will delay your circadian phase because there's actually a hormone called melatonin, which is one of the ways for our circadian clock to transmit signal in our body. And this hormone is actually kind of the preparing to sleep signal. And it is secreted mostly during dim light, in dim light conditions. So this means that if you expose yourself to bright light in the evening, you will delay the onset of melatonin production, which means that it will be harder for you to fall asleep right afterwards. So during our lifetime, sleep changes quite a lot. At the beginning, when we are really, really young, we tend to spend most of the day actually asleep. Well, most of the day, end of the night, because children sleep in really shorter bouts, as you probably have experienced if you're a parent. And during the lifetime, our sleep time decreases. And why is that? Well, because the two processes that I have explained to you a few minutes ago actually tend to become less strong with aging. So the buildup, the homeostatic buildup of sleep need is really lighter, like you accumulate sleep need a lot slower, but also the circadian signaling becomes more silent, so more dampened, and it is also slightly advanced, which is why most older people tend to go to bed and also wake up earlier. So sleep changes quite a lot with aging. We tend to sleep less. We sleep less well as well. We tend to feel less sleepy. We need more time to fall asleep. We also spend less time in deeper sleep stages, but also, and maybe counterintuitively, we are less affected by sleep deprivation because of this dampened circadian signaling. That means that we have a slightly lower sleep promoting signal and the slower accumulation of sleep need. So I told you sleep is important for cognition. So when you are refreshed, when you are well rested, you will concentrate better, you will have better attention, you will perform better at work. Sleep is also very important for memory. For instance, that's a good advice for students. If you have an exam, it's really better to study the most important things. If you're in a rush, then sleep on it and go to the exam rather than just studying the whole night, because during sleep, memory consolidates a lot better. Sleep is important for physiology as well. So sleep regulates some hormones that are responsible for the feeling of hunger and fullness. But not only. So recently, scientists discovered what they call the Glymphatic System, which is basically the brain's bin lorry. So we know that during the day, there's a lot of neuronal activity going on, which generates waste. And this waste is really cleaned better while we sleep. This is especially important because, for instance, I don't know if you have heard about Alzheimer's disease. In Alzheimer's disease, basically one of the main hallmarks is the accumulation of beta amyloid in the brain, which is also, let's say, neuronal waste byproduct, and which is cleaned during sleep. So I told you good sleep is important, but also it is really crucial to sleep at the right time. So here below you have a little graph of what the kind of data that we can have from this little piece of equipment, which is basically something like a watch. It's called an actimeter, but you can find things similar in, I don't know, sports shop. It basically counts movements. So each time you will have a movement, there will be a tiny little bar. The higher and the more movement you will do, the higher the bars will be. So if you have a healthy working clock, you can really see well the difference between here the periods of rest, because at night we are supposed to sleep, so obviously we will have less movements, and during the day you will have a lot more activity. And if everything goes well, like here, you can really tell the difference between day and night. But when you have your circadian clock that is a bit messed up in a sense, well, it's really much harder to disentangle where is the day, where is the night. And it has been shown that the higher the contrast between your day and night activity, so the less you have intrusions of periods of wakefulness during the night and intrusions of sleep between the day or rest, so naps basically. So the more regular you are active during the day and asleep during the night, the better you tend to perform cognitively. So you will have better attention, concentration, memory, and so on. Also, an interesting study showed that in retirement homes, enhancing this contrast between day and night by making people more active during the day, more awake, by exposing them to bright light in the morning and making them sleep better at night, can slow down cognitive decline. So actually, this is one of the studies that my colleagues are performing right now. And they are showing that napping can be good in the sense that if you are really tired, you didn't sleep well this night, it's good if you just go take a short nap and you will feel refreshed after it. You will be able to work better. But regular napping, especially on the long run, can actually, well, mess up completely your circadian rhythmicity. It will make you sleep worse at night. And basically, it has been shown that people who fall asleep more easily in the afternoon, so at a moment where you're not supposed to fall asleep, so those people who sleep easily in the afternoon tend to perform worse cognitively again. So I'd like to finish with a piece of advice for you. Try to keep a regular sleep-wake schedule. Also go to bed only when you're tired. It doesn't make sense for you to try to force yourself to sleep if you really feel that you're awakened and will just turn in your bed and get angry. Beware of naps, especially of late naps, which will decrease your homeostatic sleep need and make it harder for you to fall asleep during the evening. Try to keep the same wake-up time in the same rhythm in the weekend if it's possible for you. And that's really important. Go outside, take a light bath, especially in the morning, because it's really, really important for you to recalibrate your body clock. If it's not possible for you, or especially in the winter, get exposed to bright light in your office, install some good lights. This can be a good alternative. Protect your sleep environment. Try to make it as dark as possible and as soundproof as possible. Avoid stimulants such as coffee, Coke, tobacco, which will make it harder for you to fall asleep. I would even add, try to avoid alcohol, especially late in the evening, because it will make your sleep more fragmented. Favor a light supper instead of a good heavy dish right before going to bed. And also try to avoid physical activity, especially late in the afternoon or evening. Thank you. I hope you are not all asleep right now.
No, at all. It was really interesting. Thank you so much, Daphne. And I have a lot of questions, and I see that people already start with some questions in the comments. I see also Tanin that is really excited. Probably she has a lot of questions as well.
Usually, yes.
And Sebastian, if you have any questions, just make us a sign, and we are going to put you inside the conversation. Yes, he has a question.
Yeah, actually, I do. So sometimes you hear about people having these like biphasic or polyphasic sleep patterns where they sleep like an extra time somewhere during the day. And they say that really benefits them, that they actually have to sleep less in total. But it seems to completely contradict what you presented here. Is there like any scientific evidence that this is that this has any merit?
What do you mean by biphasic? Because I'm not
They
sure.
sleep in like two waves, right? So normal people, they, let's say, go to bed at 10 o'clock, they wake up at 7, and they basically break their sleep patterns into two parts. So they would go to bed somewhere during the day for a couple of hours, and then again for a couple hours during the night.
Well, I've never heard about people doing this really, like, especially on purpose. But yes, I would say that our body clock seems to be designed to have, at least in the at the adult stage, to have one consolidated period of sleep. And as I said, even if taking a nap on the short run can be beneficial, that is, I've never heard that, just dividing your sleep period in two without having any, let's say, social reason to do it could be good for you.
Okay, so you wouldn't recommend it then?
No.
Along those lines, there's another question from Guillaume Corradino. Is an individual's natural circadian cycle necessarily the healthiest rhythm for him or her, or is there a theoretical optimal sleeping habit? Is that what you described? Just stick to.
So actually, one thing that I haven't mentioned is that we all have a slightly different timing of our rhythm. So we talk about early birds and evening hours, and it is actually scientifically proven that we all have a slightly different inside time. So of course, if your work allows you to listen to your own rhythm and wake up at 9, if it's the time where you feel the most awake, then that's amazing. Of course, not all jobs allow you to do it. I have to say I have the advantage because I work in a chronobiology lab. I can usually start at any time I want. So I can arrive at 10, sleep until 8, and finish at 8 usually p.m. But so if it's possible to you to listen to this inside clock, that I think a lot of people maybe have discovered, if you have had the chance or the mishap to spend the lockdown at home, and that you could really rearrange your work schedule, then you might have realized that actually you're more comfortable going to bed at 12 or at 1 a.m. and waking up later. So if you can do it, the better for you, because that's true that someone that would, let's say on holidays, if he goes for a month of holidays, he would wake up, go to bed at 1 and wake up at 9. Well, forcing this person to wake up at 6 because that's just the job's obligations, well, he won't be this productive during the first hours. I wouldn't say it's not healthy as long as this person sleeps enough and tries to have one consolidated period of sleep, but it won't be, let's say, rather comfortable and maybe optimal for performance.
Do you know any reason why it's like that? For example, Melissa Fata asks, is the change of sleeping activity during the course of life correlated with changing in metabolism? Is there like a baseline metabolism that people have that variations, or?
That would be more a question probably for a medical doctor, I would say. So I'm a neuropsychologist. I wouldn't know the answer to this, honestly.
Okay, I have actually a questions, if I may. Just because you mentioned it, you just mentioned the lockdown. And I don't know if this is the case. Probably it's not. If it's not, just ignore my questions. But during this period, a lot of fruitful studies came out, because it was a very extraordinary situation. So there is something that actually came out during this lockdown regarding sleep habits or stuff like that. That was interesting.
So actually, what I have heard is that people tend to spend more time in bed, but or not they also tend to go to bed later than they would usually, but they don't necessarily get more sleep.
Okay, Okay.
But then
Yeah.
there's also some parts that I don't know really well, but there are some, let's say, anxiety issues as well. Especially, I think women are concerned with this, and I don't remember because it was separated into people that kept working regularly and people that were had the opportunity to work from home. And I guess those ones might sleep slightly longer, but I'm not sure it was really a significant difference.
Okay. Very good. Thank you. We have and a question just come up. Is more important to go to bed at regular time or to wake up at regular time? Tricky
Well,
question.
I would say they kind of both depend on one another. So obviously, if you go to bed sometimes at 11, sometimes at 2 a.m., well, it will be hard for you to wake up and feel well after having sometimes, I don't know, sometimes 7 hours of sleep and sometimes 4. But
so it's always better to keep the same time of sleeping instead of keeping the same time of waking up or go to bed. So it's the window of hours that we sleep the important part. That
yes,
one
I
should be
would say, well, it's more or less regular. Of course, it's almost impossible to wake up always at the same time. I mean, who wants to wake up at 7 in the weekends if there's no need for that?
Indeed.
But for instance, shift work is not a good idea, especially if you are changing, let's say, every week. So there is some flexibility. The human body is really an amazing machinery, so it can be flexible and adapt, and there's nothing wrong with I mean, it's not military, 11, 7.
Our body is going to adapt on the situation. That's,
yes, to some extent. It's just, of course, sleep deprivation and even chronic sleep restriction is not something recommended.
Okay. So we have some other questions. So Anna asks, you say that our sleeping cycle is longer than the day-night cycle. How this is possible? I would assume that the human brain tends to synchronization.
Well, as I said, and as I showed in the slide where you have really this rhythm that completely gets off, if we take you and put you in a room where there's no output whatsoever from the outside world, you have no clue what time of the day or night it is, you have no daylight, you just, let's say, you can switch on and off the light whenever you want. After about a month, you will go completely round the clock, because the first day you will go to bed at 11, wake up at 7. The next day go to bed at 11:30, wake up 7:30. This is what I mean by the human internal clock has a cycle that is slightly longer. But of course we do synchronize, because, well, it makes no sense for us to sometimes go to bed in the morning and wake up in the evening. That's what light is the major synchronizator for our internal clock to the external world. And there are other what I called Zeitgeber, so time givers, such as even just social activities. That's why we tend to keep a regular sleep-wake cycle, because, well, we are not isolated in a cave.
Yeah, definitely. So Tanina, I don't know if you have other questions.
We do, hold on one sec. We have a question from Martea, sorry if I'm mispronouncing your name, Demsar. How about blind people? Do they follow the same sleep-wake rhythm? So I'm guessing from they cannot perceive light as a stimuli.
Yes, that's a very interesting question. Actually, I know that some types of blind people that do not have, some people have still the receptor, because even when you have your eyes closed, you can perceive if there's light or not. But some type of blindness doesn't. So those people are what we call free runners. So that means that they kind of act as the people in the cave. So basically, those people really need, to have some tight schedule, let's say, for meals, social activities, and so on, because otherwise they would tend to get completely on free runner on a different time, slightly shifted each day.
Okay. But in general, blind people, they are able to have the same behavior that we have of synchronization with the light.
Yes.
In this, I imagine that are rare or at least severe case where they don't have these receptors. Okay.
Is there?
Sorry, it got really dark in here. Oh, shit.
Oops, sorry. Just a quick question, just a follow-up on that I just had. Is there any medication in the works that would help blind people like this that don't have the receptors that could imitate what?
That honestly, I don't know. I don't know what the melatonin secretion pattern of those people look like. I don't know if they might be helped with regular intake of melatonin.
Okay, makes sense. We have another question from Ali. Why is it bad if we go to exercise after 8 p.m.?
Well, I don't know if you've ever tried to stay awake the whole night. Actually, that's interesting as well, because what we do now in our labs when we do a sleep deprivation study is that we completely ban any physical exercise for the volunteers that come in. So they are basically stuck in a bed for, let's say, 20, 40 hours sometimes. They can just stand up to go to the toilet. And this is because even just moving around, walking a bit, it helps you feel less tired. So when you're driving for a long period of time, you stop at a parking and get out, walk a bit, and this helps you feel awake. For exercising, actually, if I'm not mistaken, then to fall asleep, your body temperature starts to decrease. So if you start to do physical exercise late in the evening, you increase your body temperature, which makes it harder then to cool off and harder for you to fall asleep.
Okay, good. Seems quite reasonable. So I think we have time for another couple of questions. We have these questions from Prashant. Are there any ways to improve REM sleep cycles? REM sleep cycle.
Okay, so I'm not sure what the question really means. That is something that I didn't go into. So we have different sleep stages. So we have what we call Rem and Non-Rem sleep. And REM stands for rapid eye movements. So it is also called paradoxical sleep, because actually the way to differentiate the two is to put electrodes on your scalp and register your brain electrical activity. And when we are in REM sleep, we have activity that looks similar to wakefulness. While during non-REM sleep or deep sleep, we tend to have really slower electrical waves with higher amplitude. So we do have, sleep is not a uniform behavior, let's say. And I'm not sure what the question really means. Are there any ways to improve REM sleep?
Maybe it's just referring that if there is a way to improve the quality of this cycle, because sometimes these cycles can be interrupted because we wake up, I guess. Maybe I'm saying something very...
That would be more general advice how to improve sleep, which would be don't sleep during the day, especially not in the evening. Try to have a lot of physical activity. Try to have bright light exposure in the morning. Also, well, that's more advice to fall asleep, but don't go to bed if you're not tired. So really, and keep your bed just for sleeping. So if you want to sleep because tomorrow you have to wake up early, but then you're just restlessly turning and turning in your bed, it's better to just leave your bed, take a book, go to your sofa, read a bit, and when you feel that sleepiness is coming, go back to your bed. Obviously, if you wake up several times a night to go to the bathroom, then try to stop drinking a few hours before. What could I say more?
I think that actually in your presentation at the very end, you gave some very good advice for that. I think we have time for a very last quick questions, and it's the kind that I love the most. In an alternative universe, could we have evolved as a species that does not need sleep? So you were saying that actually most of the animals and creature actually on Earth, they need to sleep. Can we imagine actually a life without sleeping?
So careful. I said that most living creatures have a circadian rhythm, which does not
okay.
sleep.
Okay. Sorry.
But to the best of my knowledge, all animals do sleep. And though it might be surprising because it's such a basic thing of life, we have not unraveled all of the reasons why we do sleep. But because of the ones that I have stated, I cannot imagine that we would be able not to sleep. Maybe in some other universe we could have really longer periods where we don't sleep, but yeah, or maybe it would just be a different state. But because of all the things that are going on during sleep, I cannot imagine that we would somehow still be conscious in the same way that we are when we're awake. I don't know if that makes sense.
Yeah. A lot of sense. And I know that there are also animals that they found other solutions, like I don't know if it's a legend or not, but dolphins, they are able to have these conditions where half of the brain is sleeping and half is awake, in a very rough terms.
Yes. So actually, dolphins are also mammals, just like us. So, and they need to go to the surface to breathe. So just to be able to, go there and take a deep breath whenever they need, there's actually, it's called unihemispherical sleep. So they have just one hemisphere that is sleeping while the other is watching guard. And it's also the case actually for some migratory birds that can fly for a
long period
in a row, and they have just some parts of the brain that is asleep. Actually, this is also, well, maybe the last thing I will say. This experiment about the guy that did not sleep for a few days, it was shown later because later experiments, they put electrodes on the scalp. So they registered electroencephalography, and they saw that it's impossible, let's say, to not completely sleep for a few days. So there are some parts of the brain that do sleep in a way, also in humans when you are, pushed to the limits. But if pushed to the limit, people and animals at least die from sleep deprivation.
Is that what happened to the guy, unfortunately?
No, he didn't. I don't think anyone has gone this far, but I know that there are some very, let's say, curious and determined scientists that prevented some puppies from sleeping, and that did kill them. So.
Oh, okay. That doesn't sound really great. Okay, Daphne, thank you so much. It was very interesting. And now we are going to have a small break before to come back for the second part with Sebastian. We are coming back at 8:30, so you have time to take another beer if you want. Daphne, you are more than welcome to stay with us to enjoy also the Sebastian's talk. And then if you have any questions for him, you can join us in the Q&A session. So I will just leave all of you with a nice set of questions, curiosity. Let's see if you're able to reply to these questions. We're just going to leave these on screen, and just before the end of the break, I'm going to project also the answers of these. These are just a few things correlated to the next topic with Sebastian. So without wasting more time, guys, see you in 10 minutes. Go to take another beer. And Daphne, thank you again for your very nice talk.
Thanks.
And here we go. Welcome back to everybody. Here with me tonight there is Tanin. Hi, Tanin. How are you? Did you
Hey,
enjoy
how's it going?
All good. Did you enjoy the first part?
Yeah, I really did. Now, I'm not for certain that I'm an 8 a.m. person when I think about it. So,
Yeah, definitely. Me too. Actually, I have to say that. So, if you just join us here for tonight, guys in the audience, welcome. This is Pint of Science 2020, and this is the third day of this special event. We are here tonight with some very nice speakers. The first one just left, and if you want to have a look of her presentation and her discussion regarding the importance of sleep and how sleeping cycles work, you should actually check our YouTube channel and Facebook page, because after the end of this event, we are going to upload everything so you can have a look if you want. But for tonight, we have another speaker with us, which is going to talk about something very interesting and very cool, something that is living in us, with us, on us. I don't know how to say exactly. Sebastian Proust, hi.
Hey, hello again.
Did you enjoy the first talk?
Yeah, it was brilliant. So a very nice talk. You can see I already put up some blue lights behind me to keep everyone awake for the next 20 minutes or so.
That was a good idea. But I think that is not going to be necessary, because I'm quite excited about your talk. So you're going basically to discuss with us regarding the importance of the bacteria and the microbes that are on our body and inside our body, and how actually we can learn more about it through the web. So something very interesting. So without wasting more time, I will just leave you the stage. But just before, I want to remember to all the audience that if you want to interact with Sebastian and with us, you can just leave a comment in the comment section on Facebook or YouTube, and we will be more than happy to reply to your question during the Q&A sessions. Okay, so Sebastian, the stage is yours. I will just leave you with your presentation. Me and Tanin, we are going in the backstage, and good luck.
Thank you very much. So before I start talking about bacteria, I'm going to show you an image that's actually from a very old scientific work. And while I give you a little bit of an explanation what it is, I would like you all to think about how this image would look if it's actually published today in a scientific journal or in a newspaper. So here it is. So this is actually an image from Isaac Newton, and it's published over 300 years ago. And this is from a work where he tries to describe all kinds of physical phenomena that appear in the world around him. In this case, it's a pendulum that swings from point B to point A and experiences some kind of resistance, some friction. Now, if this image would be published today, it would look something like this. So I redrew his image according to the guidelines that you get from a journal like Nature. And you can see that's actually pretty similar. The lines are more crisp. There's a little bit of color in there. The text changed from Latin to English, probably translated wrong because I don't speak Latin. But all in all, it's not that different. And I think that's actually a very bad thing, because the way we're doing science is not the same as people were doing science 300 years ago. For instance, we have this huge challenge ahead of us. We're generating more data than ever. So drawing figures and analyzing it is becoming more and more complicated. Just to give you one example that I made together with a student a couple of years ago is this. So that might look like a psychological test, like an ink blot or something, but actually every dot in this image is a gene from a crop sorghum. And genes that are switched on in the same tissues or during the same conditions are connected with a line together. And this is actually a very powerful tool to predict the function of genes, because a gene with a similar function is very often linked together with genes of the same function in these type of networks. However, this image is so large and so complicated that it's actually impossible to extract that information. So just publishing it for print media the way we were doing 300 years ago would not work in this case. The other problem that we're facing is that, So basically we are generating more data and it's becoming more and more complex. So it's becoming very hard to analyze. And this is actually a problem because not everyone is like a bioinformatician. For me personally, I kind of like the challenge to work with a large and complicated data set, but people have a different background, doctors, microbiologists, epidemiologists, they actually have problems working with that data. And also, if you want to reproduce something or you want to explore data sets that other people were working on, this is very difficult in this day and age. Which brings us to the next challenge. We actually need to communicate across disciplines. Science is no longer a solo sport. We really work as a team. Oops. And for this, I would like to use the Flemish Gut Flora project as an example. So for this project, we recruited 3,000 volunteers that filled in an online questionnaire, visited a doctor where they got like a routine medical checkup, and they provided us with a stool sample where then our lab technicians determined which bacteria are in there and how many of each kind. And so this is actually already a very complicated data set. So first of all, there's a lot of logistics involved to collect that data. From these online surveys, we get information about their health, their lifestyle, their diet, their medical history. The visit to the general practitioner will tell us something about their general health, their current health, but also a blood sample is drawn, and we do a simple blood test on for each participant. And the data set in the beginning is very, very complicated to analyze, right? So we don't measure the bacteria directly. We measure them indirectly through sequencing a little part of their genetic information. And to turn that little part of genetic information into bacterial abundances in each sample, you need to have a lot of bioinformatics. But as the project continues, the bioinformatics actually becomes less and less important, and the biology becomes more important. However, a bioinformatician like me might not have enough knowledge about the biomedical and the microbiology aspects to make the correct assessments or to ask the right questions. So we actually have to work with different people together on the same project. And rather than giving them a very complicated Excel sheet, which a biologist cannot analyze by him or herself, I prefer to do something else. I'd rather give them the data together with the tools to explore them in one package. And this is something that actually a lot of companies have been doing before us. If you think about Amazon, Facebook, and Google, they have a lot of data. If you think about Facebook, the amount of information that they have on you and your friends, it's enormous. And still, with a couple of clicks on a button, you can find out what your friends were doing the day before, which events they're interested in, and stuff like that. So they actually have solutions for some of the problems that we're facing, that we have a lot of information where we need to extract little bits and pieces from to answer some of the questions that we have. Now, with all these data that we collect for the VDP, I developed a platform where you can upload that data as a developer. And it presents you with a very user-friendly interface. For instance, all that metadata that comes from the questionnaire is presented in a simple list, and you can click on it to interact with it. And if you click, for instance, on gender, it will very quickly show you the distribution of men versus women in this data set. If you click on age, it will show you the age distribution for all the participants in our data set. You can immediately see that we have a little bit of an overrepresentation of people in their 50s. You would expect this curve to be much flatter in the beginning. And if you want to do more complicated comparisons, there's a button to do that as well. You could click on Explore Metadata to kind of compare one thing with the other. For instance, if you want to compare the age of men versus women, you get a plot like this. So here, the line in the middle is actually the important one. That's the median age of the men, whereas here is for the women. And you can see that while the women on the median age is about 50, for men, it's 55, 56. And when analyzing the data later on, these little differences can be important to know. And with this tool, it actually gives you a very easy way to quickly interact with the data and see these kind of changes. We can do the same thing for body mass index. So the body mass index is kind of like your weight in comparison to your length. And a healthy body mass index is between 18 and 25. And you can see that the majority of the women is in that range, whereas the men, actually half of them, the part above the median line in the middle of the box plot, is slightly obese. So you can immediately start thinking why that might be. And so one of the potential explanation I found in our data set is that men drink a lot more beer than women. So you can see here that the percentage of the men that say that they drink beer almost daily is almost 40%, whereas with women's a lot smaller. Now, these are relatively simple analyses. I'm very certain that a lot of the people watching this talk, especially the ones that have a job that involves numbers, could do something like that with Excel or an open source equivalent. However, in this case, it's so much more efficient if you can just come up with an idea, click a couple buttons, and then actually get your answer immediately. Now, once we start including the bacterial data, then it becomes really complicated, because our guts are really complex ecosystems with hundreds, if not thousands, of different species of bacteria interacting with each other. And we have to link that to the other data that we have on the participants. However, it's not because it's a complicated data set that you need to have a complicated interface to interact with it. For instance, one interface that you're all very familiar with is this one, just a search box. So the platform I developed actually has a Google-style search box. So whatever thing you can think of, you can just enter it and it will retrieve all the relevant information that we have in the database about that process or that bacteria from the database. So if you type, for instance, Prevotella, the name of one of the bacteria, you can very quickly retrieve the data that we have and start comparing the abundances of Prevotella in people with different conditions or different gender, different age, and so on and so forth. And here's one of the examples. So we measure for each sample the Bristol stool score, and that's basically telling you something about how fluid a stool sample is. And in this case, you really want this to be somewhere in the middle of the plot, 3, 4, or 5. That's healthy. If the score is lower, it's very hard. You're probably constipated. And I guess you can all guess what happens if it's above 5. So in this case, not everyone has Prevotella. That's why these box plots look a little bit weird. But the people that have Prevotella and that have a lot of it very often have problems with diarrhea. Now, I'm not saying that Prevotella causes diarrhea. We don't know that from this type of experiment, but we can very quickly pick up patterns like that and then basically postulate hypotheses that we can test later on with another type of experiment. Now, another thing that we like to do is actually zoom out and look at our complicated data set with a bird's eye perspective. So what you see here in this plot are all kinds of different dots, and every dot represents one person. And so we tried, or we ran a couple of algorithms on this data set to kind of put people that have similar composition, bacterial compositions of the gut together in this plot. And then we can start basically highlighting people based on certain characteristics. So if, for instance, we try to highlight the people that have a high abundance of Prevotella in their gut, we can see that there's like a cluster of people here on the left that appears. Now, on the right side, we have a number of people that have a lot of Bacteroides bacteria in their gut. We can also map other features that we measure indirectly, like the diversity. So that's a number of different species of bacteria that you actually have in your samples. And what you can see here is that the people on top have a lot more diverse flora in their guts than the people at the bottom. And so experiments like this have led us to the conclusion that there are actually 4 different types of gut ecosystems. We call them the enterotypes. So 2 of them basically are characterized by an abundance of Bacteroides species. One of them has like a normal amount of diversity, and the other one has reduced diversity, Bacteroides 2. Then that cluster of people that have a high abundance in Prevotella, that's another enterotype. And then the ones that have very high diversity on the top of that plot have a Ruminococcus enterotype. And so what we know is that the Bacteroides people actually very often have problems with inflammation in the gut and other types of problems, even psychological problems. And one of my students wanted to see like, okay, is this like related with medication? Can I link this with antibiotic usage? And something interesting appeared there. So here we're trying to link some of the medical data that we have on these people with the gut microbiome. And so what he did was he looked at the antibiotic consumption before they took the sample, grouped all the samples in people that had antibiotics 3 months, 3 to 6 months, 6 to 9 months, and so on before taking the sample, and then looked at the distribution of the enterotypes within those groups. And what you can see is that Bacteroides 2 enterotypes actually take antibiotica much more frequent in recent months than you would expect to, or when compared to people that took no antibiotics in the period leading up to taking the sample. Now, again, this is a correlation. It's not a causation. So we don't know if Bacteroides 2 is caused by taking antibiotics, or if people with a B2 enterotype in general have bad health and just need antibiotics more often than other people. But we could design other experiments to test for that. Now, when working with these data sets, we can also find out things about those bacteria. If you think about Facebook again, they try to find out something about you based on the connections that you have with other people. Most likely, you will like things that your friends like. That's kind of similar to what we're trying to do with bacteria. So we can see which bacteria very often occur together in samples and which ones are actually actively avoiding each other in samples. And so if they are friends, between quotes, we can draw a red line in between them. That means that they co-occur in samples. The ones that tend to avoid each other have a blue line drawn in between them. And this is something that can help us a lot to kind of figure out how these ecosystems work, because the ones that are connected are like kind of little consortia of bacteria that might actually need each other to function well. And again, this drives the experiments that we do in the lab. So in a nutshell, the tools that I've been developing are very easy to use, which means that more people, also people with a non-bioinformatics background, can actually leverage this data. And they're very efficient to use. It's very easy to retrieve the information that you need, so you can get novel insights faster. Now, unfortunately, you can't play around with this platform yet because it hasn't been published and it is not publicly available yet. But if you want to see some of the concepts that I've explained today in action, there are websites where you can do this. So, former collaborator of mine, Thomas, who just got his PhD a couple days ago, so congratulations to him, made this image for a publication. And it's a very complicated thing, right? It's about genome evolution of a specific type of genes in plants. And he made this image as part of his PhD. And you can see it's very information dense. There are lots of colors. There's a lot of different things going on here, which makes it actually very difficult to interpret. So when we tried to publish this image, one of the reviewers said, like, yeah, it's too difficult. I can't get the information out of it that I need. You need to find a solution for this. And it's very clear that the amount of information we have in this image cannot be captured in a single image in print media. So what we did was we made an interactive version of it. And this one you can find on the URL shown below. And basically there, you can start to click these nodes to get the information. You can drag and drop them to kind of like see if there's something interesting below them, right? So that you can click also these edges below them. And you can really like explore this visually much better than you can do with a single static image. Similarly, we had a lot of like phylogenetic trees that kind of like give you the relations between genes in this gene family for all these plant species. And they were basically impossible to read because they're very, very large. So we used some of the technology that's similar to what Google Maps, for instance, is using to make these images dynamic, that you can zoom in on them indefinitely and that you can really find the information that you need. Now, if you don't care about this type of genes or genome evolution, there's another one that I think everyone will know. It's this dashboard that Johns Hopkins University made. And this is actually a very good example of what I've been explaining, why we need these kind of like tools to play around with data. And even though the data here is absolutely terrible, right, and it's forcing me to work at home for the last 6 months, the concept is kind of great. because we have a very complex data set. It's time resolved, it's geospatial, yet Johns Hopkins University was able to make an interface that everyone can use at the click of the button. And I really think that we need to go to this type of system much faster and much more often than we've been doing now, because it really helps us understand what's going on with certain data sets much, much better. Okay, so I'd like to thank you for your attention. If there are any more questions, I'll happy to take them.
Thank you so much, Sebastian. It was very interesting. It was also very curious to see that there is so much about this, and it's also really nice, as you can see, some similarity with words like Facebook. So the social media words, actually, it's something that we had the idea that it is a waste of time, but in the end, it's a model that we can apply with something more and more interesting.
There is actually a lot of interesting open source software that's emerging from Facebook. You can complain about the privacy of the platform in general, but some of the technology that they're developing is actually really helpful for me to do what I want to do.
Indeed. It's again this endless debate on how you can use the web in a correct way, instead of just making money, but without real, maybe sometimes real ethic behind. So I invite all the audience to ask questions if you have any. And also, Daphne, if you have any questions or you want to join the conversation, just make us a sign, shake your hand, so we know that you have some questions. You can also join us later if something come up in your mind. We will keep an eye on you. So if you want, you can join. Tanin, I don't know if you have any questions.
Actually, I do. So, Sebastian, thank you so much for the talk. It's really fascinating to see how we can start looking at complex data sets. I was just wondering, do these interactions have any clinical relevance? So right now, I guess this data set would be useful for research in terms of looking at novel or unanticipated interactions. Is there any clinical or practical basis in a non-research environment?
So there are plenty of websites that claim that they have a solution to improve your gut health. The vast majority of that has no scientific proof yet. I mean, it is clear that there are links between our well-being and what is happening in the gut. But I also stressed this during the presentation, the correlation is there, but that doesn't mean that one is causing the other, right? Mm-hmm. However, it is a very interesting target, because if the gut is causing something that makes you unhealthy, it is potentially something we can change, right? Mm-hmm. It is possible to get the bacteria out of your gut and put other ones, potentially better ones, in. So there are definitely studies going on trying to treat certain diseases, transferring fecal matter from one patient to another one, or trying to change your diet to kind of modulate what's happening in the gut, right? But a lot of this is still very early on, and the clinical applications are not there yet. They might be there in a couple of years, right? But that's you need a glass globe to figure that out.
Okay, very, very nice. Thank you. And I have actually a question regarding how to obtain all this information. So all these softwares that actually are in development, they need a source of information. And sometimes this source of information is not free. So do you think that all these instruments can benefit of a free information world where actually you can share all the data between different, research centers or companies or universities and something like that? So is this very important for this kind of project that you are building on?
So for several projects, especially what I've been working on in the past with comparative genomics, there we relied solely on publicly available data.
Okay.
So I would not have been able to do my PhD without the access to publicly available data sets. That being said, I feel very strongly about this because a lot of science is funded with taxpayers' money. So I think that if people invest in this through paying their taxes. Also, something should come back from science to the public domain, right? So by making your data public, that's actually what you do. And by making it public, you allow other people to work with it and do other things with it, maybe things that you hadn't envisioned in the beginning, which is also good, right? So it kind of like increases the return of investments from the taxpayers, which I think is a very good thing.
We have a comment from Bart Smith, and he says that clinical data falls under GDPR regulation. So is your public data set then limited to other types of privacy-friendly data sets then?
So especially when medical data is involved, we need to adhere to very strict rules, what we can and cannot make public. That's something we're very well aware of. It's also something that we're very careful with, right? So with the Flemish Gut Flora project, we'll have to make sure that what we make public is possible to make public. So there are some privacy concerns, and it's something that we really take into account. It's also something that we're not, like if we don't have a legal basis to make it public, we would never make it public, right? In a lot of cases, the data is available then to collaborators, to scientific collaborations, but then it's not something we could just throw out on the internet. That's absolutely true. For medical data, that's a concern.
We have another question from Bart Smith, that he learned a few years ago that there was a relationship between gut flora and obesity, and he wanted to know, what is the status of this topic?
Well, so as you might have guessed from the presentation, I'm much more about the bites than about the bacteria, right? But we do see that there are some relations between obesity and gut flora. There was also a recent publication where I did a tiny part where we could see that basically people that had a different gut microbiome and that were taking certain medications were actually better off, even though they were obese, when they were on that medication, even though it was not doing anything for specifically for obesity. And that might be through an interaction with the gut microbiome. So this work is definitely ongoing. I think it's all very interesting to find out these relations between different aspects of our health and what's happening in the gut. But it's still early days, right? So it's a bit early to really say like, okay, this is like what's going to happen. It's very, very early days for that.
Nice.
Okay, we have another question from Prashant. He is asking, I have a question regarding the antibiotics experiment. Did you take into account various types of antibodies and route of administration, as that might impact your data?
We don't know. The student will come back for a master thesis next year, and that's his job to find out.
Okay, so it's still going on, basically. Yeah. Actually, I have questions. I think we have also questions from the audience, from Lucas. Besides the very nice figures, how far will this platform be useful for advanced stats?
Right. So I consider this very much something that's exploratory, right? So it can help you find a specific thing that you're interested in and generate a hypothesis. But then exactly which stats you should use to prove that point is usually very, very specific to your problem. So it's difficult to kind of like embed that into a generic platform like this one. So I do have options pretty much on every page that allow you to download the data and just load it into R or in Excel or whatever program you want to do those more advanced statistics. But I don't do that much statistics on the site here. However, there are certain tools that you can use to basically compare two groups of people properly statistically, and then import the data into the platform and start basically including that in certain images. So the tool I developed is more for visualizing and exploring the data set. The stats I really basically let other tools handle or put it into the things that the user should kind of like do later on to really verify everything.
Okay, great. I have a question, actually. So you showed the very nice platform from John Hopkins regarding the COVID situation, the COVID emergency. and it has a very, I would say, social application. So it's something that for citizens is very important to track this, and was very helpful. And do you think that also these platforms regarding bacteria can be also used from normal citizen, or is something that is mainly related to doctors or scientists? Because the risk is always that, as Google became your doctor, also this platform that can become your doctor. So is there something that you thought about it, or is something that is just...
Well, I think we should probably put like a big disclaimer on there that like, if you are sick, you should go and visit a proper doctor, right? And not kind of like use our tools to start and treat yourself. However, I do think there's merit in having these tools available, right? It really democratizes science. Because if I would release this data or the data from Thomas's paper just as raw data, you're going to spend weeks trying to do something with it. Yeah. And the chance of someone doing that is very low. Whereas if it's released together with some tools to play around with it, people probably will, right? Or at least it decreases the chance that someone will actually have a look at it, and maybe they find something, maybe they don't. But the option exists without spending weeks or months of coding to kind of come up with something yourself.
Yeah, definitely. Probably we should invest a bit more time and money, not just in produce results, but also to produce data, but also to learn how to interact and translate this data in fact and in pure observation. I want also, keeping on this topic, do you think that all this can be also part of a project of citizen science, in the sense that you can involve citizens to help you to collect data, maybe through some gamification of these projects, so to be able then to have people that basically they bring to you informations that you can use in your database, or is something that should be more pure based on data that you collect from labs, from all this stuff?
Michele, are you asking people to go around collecting poop?
I'm not just saying that. It's not what I'm proposing, but this is something that you propose right now. So I'm not saying that. But no, it's just this. So do you think that this is something that an involvement from the citizens, from the community, can be a good input, or something that actually doesn't really matter for this type of project?
Well, in this case, we're already relying on the goodwill of 3,000 people to provide us with a sample, right? Basically, we just put a call out, then see who responds, and people don't get anything in return for it, right? So they really do this because they are interested in our science, and they just want to help us further along. Which is super nice, right? Because it's not the most fun thing in the world to basically take a stool sample and ship it to us.
Yeah. No, that's for sure. That's for sure.
And I think actually that if you could release a platform like this, also of course taking into account GDPR and privacy concerns with the data, but you can show that something is being done with it and it leads to something, is very nice, right? Because people do this out of the goodness of their heart. They're interested in science. They want to help us along, right? So if they can see that something is happening with this every once in a while, that might convince more people to join future studies. So I think there's a lot of value there as well. If we can turn this into a video game where people can try to analyze something through solving puzzles, I don't see like an immediate application there. I think it's for finding tumor cells or something from certain spectra that they have something, right?
This was in fact, this is exactly the question from Renata. She is asking exactly if this platform can be transformed in something that can be more interactive for people, just classic, except for instead of classic visualization of the data. So something that is very interactive in this sense. So I will continue with another question. If Tanin, you don't have any questions while we're waiting for others from the audience.
I cannot deny you the opportunity to ask, so go ahead.
And I was thinking actually. You were saying something funny, and that this pops up in my mind. People just send to you a sample of their poop to have an analysis, something like that. Which reminds me what is very popular lately. This kit where actually you take part of your DNA for have a sequencing of your genome to know your ancestors and stuff like this. That apparently is not very, very precise. Do you think this is some service that in the future might be interesting to understand, where we are going to have more knowledge about the microbiome of our body to, have like an understanding of what is in our body and how these things that are in our body can impact our everyday life or our health and something like that? Do you think it's something that is possible in the future?
It's actually something that's available already, right?
Oh, wow.
There are some companies that are offering this service. So I think for like 160 euros, you can send them a sample and they will kind of give you a report about which bacteria are in your gut.
Okay.
There was a documentary about the VRT, and they contacted us also to kind of like have a look at what we could learn from a sample, and also what that report that you would get in return from that company looks like. And it's actually a bit risky. Right, so they will tell you like, okay, for instance, Prevotella, you have this much, it's more or less than the average. That's okay, right? But then they also try to put like a health score on this, and that's where it starts getting risky, because all of this is very anecdotal. Like I said, it's early days, right? And for instance, in case of that reporter, the report he got back was actually pretty bad, right? Because he had low diversity. But that's not necessarily a problem for your health, right?
Or at least we don't have the knowledge right now to understand if it's really a problem. Okay.
We saw that like if you are a Prevotella type, you can have low diversity or lower diversity than average, but usually those people report excellent health. They feel better than other people, right? So it gets very fishy if you start like converting this into a health score and then giving people advice what foods they want to eat. So like you're probably giving them more data than what you're getting back, right? So because they of course are collecting data on you, the same with like the genomics tests, and it's a bit questionable what you get for your money right now. So I would argue against this, right? Maybe for some people it's interesting to know, but be careful with the information you get back. It's all early days. It's not, a lot of it is still not scientifically verified. So you have to be careful what you do with the data, and you have to think critically about what's happening with the data that you give, right? So we are open about it. We have an ethical committee that's backing us, and we're making sure that we're doing everything according to the letter of the law. Are those companies doing that as well? We don't know.
Yeah. We need to be careful as always with this kind of data. That's for sure.
Yeah. So because we're still in early days with this kind of research, could you tell us maybe what is the point that we can confidently start saying, yes, these associations that we see have some metal to them, that they hold water, that you can confidently say, I see this particular ecosystem type is really linked with something that I should be concerned about, and I should possibly consider some consultation. When would that point be?
So like a lot of the studies that we've been doing now, they're very broad. We try to sample a lot of people and see what's kind of like going on there. And it allows us to pick up those correlations. If you want to basically find a causation, right, you need to follow certain people for a certain time and then see if something in the gut changes and then later on changes in their health. Or you have to do an intervention study where you basically start following people. Some people, it's one point, you give a high dose of antibiotics to kind of like destroy a lot of things in their gut and then see what happens with both their health and their gut. For sure, those kind of studies are going on for certain types of things.
To do that, though, with giving a high dose of antibiotics to destroy the gut?
Again, I'm more about the bites than about the bacteria.
Don't try this at home, guys, if you don't like it.
I wouldn't sign up for that study.
Yeah.
Especially since we know that P2 kind of like occurs more frequently in people that took antibiotics recently, and we know that B2 is cool. So I would be very careful with that. But so, I mean, it's an extreme example, right? You can also come up with less extreme examples where you just change food patterns for a while, see what happens with their health. So those studies are for sure going on, if not in our lab, then across the world, right? But yeah, it's hard to say, right? So maybe in 5 years we will have something. Maybe it will be 15 years, maybe never. It's a very hard question. I know everyone wants to get advice on what they should eat, right? But so right now, there's not really a lot that I can say about it with certainty.
So don't click on the Facebook ads with all the happy advice on what to do.
No, I would be careful with those.
Yeah. We need to be careful with this, but probably in the next years we are going to have more and more information about the microbiome, and it's going to revolutionize probably biology once again. And so we don't have any other questions. So thank you so much, Sebastian. It was a very, very interesting talk.
Thanks for having me.
And I will just invite Daphne back on stage just for the wrap up and for the final greetings. So, guys, it was really a pleasure to have you both here with us. I hope that you enjoyed to talk with this while we are in our couch drinking beer. It was really, really, really nice to have you. I hope that you are going to follow our next event, and I hope that we are going to see each other maybe in the live events in Leuven or in other cities, in Liège or wherever, when we are the next year, probably, if we have the chance. I'm sure it's going to be the case. And so I will just first say hello to our guests. And that will take a bit of time to advertise for the next event of Couch of Science. Thanks again, guys. You can just leave us and go to have dinner or to enjoy another beer, even though it starts to be too late, as Daphne said. So don't drink too much after a certain hours, otherwise you are not going to sleep very well. And that's it. So I hope that you enjoyed. Thank you again also to you, Tanin, for your help, for your support. Thanks to all the guys in the backstage. You are the best guys. Thank you for everything. And that's it for tonight. We have done. Thank you so much.
Yes.
Thanks for having us.
Thank you. Thank you. And have a nice evening, guys.
You too.
Sebastian.
So just to wrap up for the very final part of this discussion, tomorrow we are going to have another event. Will be a very special event because we'll be live, so not online, but we'll be with real people. But they cannot touch so much each other. So we are going to have anyway some distances and to be safe. Some of you probably they already have the tickets. For the rest, probably is not the case because it's sold out. However, you have the chance to see these events in our YouTube and Facebook page in the next days, because the idea is to upload after. We want to have a very nice quality of recording, so that's why we are not going to do this live. But the event is going to be online in the next days. So subscribe to our Facebook page and our YouTube channel to be aware of this. And of course, also on Instagram and Twitter, we are quite active also there to see all our activities. And the event of tomorrow is regarding the shaping of the city of tomorrow. There are 2 different talks, very interesting, and also a interactive game / opportunity to share with the rest of the person there, which is your ideas for better cities. And so I hope that all of you can check on YouTube and Facebook to be aware about this and see and let us know if you have any feedbacks. In October, we are probably coming back with other Couch of Science episodes, which is more or less the same structure that you've seen here, but a bit shorter, more compact, to be lighter for people. We can do this Pint of Science once in a while, but do every time it will be a bit heavy, and will be probably too much beer to have all together.
Well, drink plenty if it's lighter. So
Yeah, exactly. So once again, I just want to say thank you to Tanin and all the other guys in the backstage. Thank you so much, guys, for your support and for your help. And I think that's the time to say bye and see you each other probably next event in I don't know, in Couch of Science, or maybe we are going to see each other live tomorrow at La Tricoterie in Brussels. Thank you so much again to everybody.